Hi everyone, welcome to my channel Pure Biology, and today I come with another video. But before going to the next video, I have to say something that in my previous video I just explained only the theoretical part, how the spliceosome is formed or splicing apparatus that is formed, but just the video was too length, and that's why I didn't explain about this process diagrammatically. Okay, so that's why here in this video here I try to explain another topic. So before going to the second topic, I just explain the spliceosome formation diagrammatically. Okay, so this is the spliceosome formation, or you can say that the splicing apparatus formation. So what is that? First, you can see these two are the exons, okay, and these two are the introns. And in the first step, you can see that this BBP protein, and here I just explain this in the short name wise, because there is very less time, because another topic I have to explain. That's why I just explain this diagram in very short form, and you just check out my previous video where I explain what is this BBB and BBB protein and U2AF, what are that. You just check out for this complete explanation in my previous video. So first U1, that is the protein, and BBP, this is another protein, and U2AF, this is the factor. Okay, so here the A is the point where BBP protein that bind here, and U2AF factor is there, and U1 is there. So in the next step, when the E complex is already formed, in the next step you can see that this U2SNRNP. And what is SNRNP? For knowing this about SNRNP, so you have to see the previous video. This SNRNP, U2SNRNP, that bind with the A, and BBP protein that has already bind with this A, this is just released. Okay, this is just released. So what happened here? That U2SNRNP that is bound with this A point, and U2AF is already there. And in the next step, you can see that U6, U4 and U5, these all bind with this A point, and obviously U1, this also bind with this A point. That means U1, U4, U2, U5, U6 all form a complex, and this U2AF factor that is released from here, and in that way the B1 complex is formed. Okay, in the next step you can see that U1, this is released, okay, from the complex, and so which are already bound that region, this U6, U5, U4 and U2, and here you can see a loop-like structure is formed. Clear? And in the next step, U4, that is also released, so only U6, U5 and U2, these remain bound there. And here you can see this is formed like lariat structure, and you can see that this lariat structure is released in the next step, and these exons that are bound with each other. Okay, so this is the splicing process is completed here, and you can see that E complex first converted to the A complex converted to B1 complex, B1 complex converted to B2 complex, and B2 complex that is converted to the C complex. And now this structure is looking like a lariat structure. This is totally released from the with the intron region. These two are the introns, these intron regions, and only remain there 5' exon and 3' exon. So due to the releasing of this complex with these introns, the 5' exon and the 3' exon both are come too close together, and that's why these exons are attached to it there and to form a complete mRNA. Clear? So This is just diagrammatically explanation of the splicing apparatus. And in this video I will try to explain about the splicing process, obviously, but the main splicing process I have explained. Here another two types of splicing process are there, so I will try to explain that one. Okay, so continue with me. Okay, so here this is the alternate splicing and this is the trans splicing. The alternate splicing is what? Here the alternate splicing is that only one mature mRNA is generated from a single pre-mRNA in all tissues. But in some cases, however, in case of alternate, the 5' or 3' splice sites are used during splicing. So what is saying that? In case of general splicing process, what is happening? The exons are ligated and the introns are separated from each other. And in that case, a single pre-mRNA is formed, or you can say that single mRNA is formed. But in case of alternate splicing, here the 5' sites or 3' splice sites, these are used during this process and resulting in the production of more than one mRNA species from a single pre-mRNA. And the production of the different mRNA products or RNA products from a single product by change in usage of splicing junctions. So what is saying that in case of alternate splicing, that means the more than one RNA or mRNA, full mRNA that is formed from single pre-mRNA. In general cases, there is only one mRNA is formed from the pre-mRNA because exons are added here, introns are just released. But in case of alternate splicing, the more than one mRNA formed from the pre-mRNA. Clear? So this is the actual process or alternate splicing process. And here the 5' or 3' splice sites, these are used for the splicing process. Either 5' and 3' both are used, or 5' or 3' that used in the separate way. That's why more than one mRNA formed. Clear? The utilization of the alternate splice sites can result in structurally distinct mRNAs by either excluding potential exon sequences or incorporating non-coding intron sequences. Okay, so this is happening here. That means either exons excluding of the potential exon is seen here, or incorporating of introns sequences. That means both are possible. If exons may be included or excluded, or in case of introns they may be incorporated, rather exclusion. Clear? So this is the alternate processing, and this is not regulated sometimes by the cell, but sometimes this is regulated in the tissue or according to the cell it is happening. So this is the alternate splicing. What is the trans splicing? Trans splicing is that exons from two separate RNA transcripts that are spliced together to form a mature RNA molecule. That means two RNA involved here, and from the two RNA exons are added and form a full mature RNA. This is the trans splicing. And here the 5' end of many different transcripts produced by the cell. In this way, all products of the trans splicing have the same 5' exon and different 3' exons. So what is saying that? That is saying a single exon is spliced onto the 5' end. Single exon is spliced where? Onto the 5' end of many different RNA transcript. That means only one point, that is the 5' point. This is important for the splicing process. So in the all over, like that if the two RNAs are involved, so only the 5' end of the both RNA, the place where the splicing is occurring. Okay, so that's why in that way the products of the trans splicing have the same 5' exons and different 3' exons. So all the 5' exons are same, but 3' exons are different, because the splicing started in the different mRNA or RNA. That is, the point is the 5' site. And the two exons initially found in the two separate RNA molecules are spliced together into single messenger. So from one RNA molecule, one exon is attached with the other exons that is coming from the other RNA molecule that is attached. Okay, so that's why two exons initially found here, that is coming from the two separate RNA and together into the single messenger and messenger RNA. But here one different thing is that the structure of the spliceosome generally in general splicing process, that is the lariat shaped, but here the spliceosome that is formed is in the Y shaped. Okay, why this is occurring? Because the initial reaction brings together two RNA molecules rather than forming a loop within a single molecule. So general splicing, that is occurs in a single mRNA molecules or RNA molecule. So but here the two RNA molecules that are involved, that's why so loop is not formed here. There is one exon coming from one RNA and another exon coming from another RNA. So that's why no loop formation is needed. So here you can see that Y-shaped structure or Y-shaped splicing apparatus. Clear? So this is the splicing process I have explained clearly, and I request you, the newcomers, mainly newcomers, please subscribe this channel if this is helpful to you, and share obviously, and just hit the bell icon for getting all the notifications of my uploading videos. Okay, so thank you.